So since humans really began to observe the world around them, they understood that traits passed on to offspring. They knew that their children looked like themselves and that their animals looked like the animals that they had before. So that's the idea of heredity, that stuff is inherited or passed on. But there was really two major ideas in heredity in the 1800s. And that was either the blending hypothesis or the particulate hypothesis. The blending hypothesis saw heredity as more like paint. So the genes from one parent were maybe one colored paint, maybe blue paint, and the genes from the other parent was yellow paint. And so if you took blue paint and yellow paint and mixed it together, you'd get green paint. But just like paint, that's inseparable. So then that individual that was mixed, that green color, could only pass on green because you couldn't separate back out blue and yellow paint from that mixed green paint. The alternative hypothesis is the particulate mechanism of inheritance. And this sees genes and genetic material as distinct particles, so that's where this term particulate comes from. So instead of having paint, you have two distinct objects. And they're either, in this case, I have a pink piece of paper and a yellow piece of paper. And so each individual gets those two pieces of paper. You might only be able to see the pink color, right? But the yellow is still there. And so this individual, when they go to give their genetics to their children, can separate these and give pink or yellow to their child. So parents pass on these discrete units. It's not a blended, like paint, mechanism of inheritance. So that is what we know to be true today. And the person who really demonstrated this is a very famous guy in the story of biology. His name is Gregor Mendel. And he used garden peas to study inheritance. The reason why he used garden peas was that there's a lot of traits that can just be easily seen with the garden pea. And also, they're normally self-fertilizing. So that means that they normally breed with themselves. But it's really easy to go ahead and make crosses, so to cross two different flowers. So it's both the default is self-fertilization, and you know that if you're the experimenter. Otherwise, you can intentionally cross-fertilize them. So these are the garden pea traits that he studied: flower color, flower position, seed color, seed shape, pod color, and height of the plant. These are traits, or also called characters, and he was going to study all 7 of these. So remember I said that the garden pea can self-fertilize, so it has a very specific flower structure. So you can see the petals like this, they're enclosing. So normally, as long as the petals stay closed, the male parts, which is the stamen here, and the female parts will self-fertilize. So the flower has both male and female parts, makes both sperm and egg, and it will self-fertilize as long as nothing is done. Because of this self-fertilization, you can have a flower or a variety that is what's called true breeding. That means if they self-fertilize, they always produce the same trait. So for example, this flower is white. If it's true breeding for white flower color, then its parents were always white and its offspring will always be white. So true breeding is when it displays the same trait over many, many generations. Cross-fertilization can easily be accomplished with the garden pea by taking the male pollen from one flower and putting it onto the female part of the other flower and removing the male parts from that same flower. So this is a hybridization. It's taking two plants that differ in a trait and crossing them. So in this case, it's taking a purple flower and a white flower and cross-fertilizing them. So he's going to utilize both self-fertilization and cross-fertilization in his experiments. So the first experiment he did is called a monohybrid cross. This is when an experimenter only follows one trait, so that's where the name monohybrid comes from. So it starts with the P generation. So the P generation stands for parental generation. It's like the grandparents in the experiment. And he takes 2 true breeding parents. So this is a true breeding tall plant and a true breeding dwarf plant, and he cross-fertilizes them. Then he plants those seeds and he gets his F1 generation. So the F1 generation is the offspring of the P generation. F1 stands for first filial. Filial means son and land. So these are the offspring of the P generation. And what he observed was that all of these offspring, all of the hybrids, because they're the result of a cross-fertilization, they're called hybrids, all of them were tall. So it seemed like the dwarf trait had disappeared. He then allowed the tall hybrids of the F1 generation to self-fertilize. So now not doing a cross-fertilization, allowing them to self-fertilize, and then he planted those seeds and looked at the traits of the offspring. And the F2 generation is now the offspring of the F1 generation. And now he saw that the majority of the plants were tall, but a small number of them were dwarf. So the dwarf trait had reappeared. And they had reappeared in an approximate ratio of 3:1. A 3:1 ratio, if you're not familiar, is the same as saying that 75% of them were tall and 25% of them were dwarf. How did I get to that? Well, 3:1 is 3 out of 4 total, because 3 plus 1 is 4, and that's 1/4. So 3:1 is saying that 3/4 of them were tall and 1/4 was dwarf. So he repeated this experiment for every trait that he studied in the garden pea. And every time he did that, he saw the same pattern. In the F1 generation, one trait disappeared. So if I look at the flower color here, he crossed purple and white flowers. In the F1 generation, they were all purple, so the white flowers had disappeared. But then in the F2 generation, the white flowers reappeared, but in a small number, so at approximately a 3:1 ratio. And again, he did this for all of the traits he was studying, and he always saw the same thing. One of the traits would disappear in the F1 generation, and it would then reappear in the F2 generation with this 3:1 ratio. So from his data, he concluded 3 major things. First of all, he observed that there seems to be a dominant and recessive relationship between the traits. He called the dominant trait the one that appears in the F1 generation, and the recessive trait the one that disappears in the F1 generation. So if I go back to this data, the purple, in the F1 generation, we only saw purple flowers. So purple must be dominant, and white flowers must be recessive because they disappeared in the F1 generation. Same thing with yellow and green seeds. Yellow must be dominant because I see it in F1 and green disappeared. Oops, wrong direction, sorry about that. Okay, so the next thing that he concluded is that the genetic material is made up of these genes, and genes are present in different forms called alleles. So for any trait, so for example, for flower color, so if my trait is flower color, there's a gene, and every flower gets 2 copies of that gene, and there's variants of that gene that are called alleles. So for flower color, the 2 variants in the garden pea are purple and white. So the gene has 2 variant forms called alleles, the purple allele or the white allele. In genetics, we use a sort of shorthand for the alleles. So we always use the letter of the dominant trait. So the letter of the dominant trait, or the dominant allele. So in this case, purple is dominant, so we're going to use the letter P. If we're representing the dominant allele, then we use a capital P. If we're talking about the recessive allele, we're going to use a lowercase p. So you need to be really careful in distinguishing your lowercase and uppercase letters. So use like a little curly Q, put a line underneath so you can tell you meant an uppercase or lowercase letter. Believe it or not, I've seen people mess up these kinds of questions on exams because they couldn't read their own handwriting. So be really careful about telling apart these uppercase and lowercase letters. Okay, so we have every person has 2 genes for a particular trait or 2 copies of the gene. The genes can have variant forms called alleles. All right, his third big piece that he concluded from this is actually so important. It's called the law of segregation. And it is that those 2 copies of a gene that every organism has will separate, those 2 copies will separate from each other. So that's what segregate means. That's where it's called the law of segregation. So that each sperm or egg only carries 1 allele. So for example, you might have a plant that has 2 big P alleles. So remember, it has 2 copies of the flower color gene, and this one happens to have 2 of the purple alleles. If it's a female and it's going to make eggs, then it's going to separate that so that each egg only has 1 allele. So every gamete, egg or sperm, will only end up carrying 1 allele, and that's Mendel's law of segregation. So a couple of other terms before we look again at Mendel's cross. The genotype refers to the allele makeup. So when we're talking about whether they have the purple allele or the white allele or the tall allele or the dwarf allele, we give letters, and that's the genetic composition. So I said, this is a genotype, this is a genotype, this is a genotype. Anytime you have 2 of the same, that's called homozygous. So this is a homozygous dominant individual, and this is a homozygous recessive individual. Anytime you have 2 of the same, they're heterozygous. The phenotype is the physical characteristics. So an individual that has 2 big T alleles or a big T and little t is going to they're both going to look tall. So they both have the tall phenotype. Only an individual with the 2 dwarf alleles will have the dwarf phenotype because it's recessive. Okay. So genotype is the genetic composition that's going to correlate to a phenotype. So in this case, the heterozygote and the homozygous dominant individual will be tall, have the tall phenotype, and the homozygous recessive individual will have the dwarf phenotype. So if we look again at Mendel's cross, we see that in the F1 generation, well, let's do the P generation actually first. So let me add in here the P generation. We took 2 true breeders, a true breeding tall crossed with a true breeding dwarf. And what true breeding is really telling us is that the genotype is homozygous. So a true breeding tall plant must have 2 tall alleles, and a true breeding dwarf plant must have 2 recessive alleles. So when they go to make egg and sperm, this one can only make egg or sperm with the big T. This one can only make egg or sperm with the little t. So no matter which combination you get, they're always going to create heterozygous individuals. So the F1 is all heterozygous, and that's why they all appear tall. This is showing us the F1 generation. So remember, this is a self-fertilization. So it's a big T, little t crossing with a big T, little t. So we're drawing it out this way even though it's a self-fertilization. So because they're heterozygous, they can either have a big T or a little t in their gametes. So this one can have either a big T or a little t, and because this is the same, it also either has a big T or a little t. And so then you do all of these possible combinations. So big T with big T is going to give you this one. Big T with little t, whoops, sorry, that's not right. Big T with, I did that one already. Big T with little t. So you can follow their arrows too. Sorry, I kind of made a mess there. So big T with big T or big T with little t is this one. And then this one combines with this one, gives us another big T, little t, or this one combines with this one and gives us a little t, little t. So these are all the different combinations you can get from these different gametes. And you see that you get 3 tall offspring, so all of these will be tall, to 1 dwarf offspring. So that matches Mendel's data. Every individual has 2 copies of the gene. They separate in the egg and the sperm. and then you have this dominant or recessive relationship.